Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Center for Mesoscale Mapping, housed in the Martinos Center. It is my great pleasure today to introduce our speaker today, Dr. Ann Berry. Dr. Ann Berry is an assistant professor of psychology and neuroscience at Brandeis University. Her research focuses on understanding the role of neuromodulator systems in age-related changes in cognitive functions. I would like to remind our audience to address any questions using Q&A box or raise your hands at the end at the top. Dr. Berry, thank you very much for coming today, and the stage is yours.
All right, thank you so much for having me. Just a warning that this is a brand new computer, and for some reason I can't hear questions from the virtual audience, but we have a computer here that a normal computer with normal auditory functions, and then there's always the chat. But I encourage you, if you have a question, to interrupt me and feel free to ask. And then part of my graduate training was in a very intense, I said toxic earlier, but that's not really what I meant, really intense sort of training ground. So if things get derailed, I could get myself out of it. So don't worry about asking too many questions. Okay. So my focus is, I'm a cognitive neuroscientist, but my interest has always been in the role of neuromodulator systems. So in grad school, I studied the cholinergic system, and really now I'm more in the world of catecholamines and monoamines. Let's see. Okay, and that's working. Okay, so how do we, I don't know if I can move this to get it out of the way, or if it's just always gonna be there. Maybe more. It's not showing up here, but I can
go to more. Yeah, it's usually more and hide it. Could you get to the more?
It's not letting me. I can also just read the titles of each of my slides.
I could could you with this computer, could you not do the similar screen but or extend the
Yeah, I don't need like viewer or whatever.
If you can do this extended screen, we can just put it on the
okay.
Second.
Do you know how to do that?
In the Mac, you need just to get to the screen settings. I don't know how to do it in the Mac. Do you know how to do it in the Mac? Turn the screen settings. You just need to extend.
Like just try adding more. Yeah, more here.
Okay, here you can just optimize, hide video panel.
Hide video panel.
Start sharing the one below it. Floating
Meeting controls. This one.
Hide floating meeting controls. Okay, cool. And then
press ESC to show. So like press ESC and share it.
I think it's okay, right?
No, we should share the screen for just do the presentation.
I think you just do F5 for presentation.
Present our view.
Yeah.
Can the online people still see the slides? Okay, cool. All right, take two. Thanks. The joys of the hybrid. Okay, so my lab uses, so I study humans. And so we use a number of different imperfect tools to get at the role of neuromodulators in cognitive function. So pharmacology, we have also used looked at different genetic polymorphisms inferred to affect neuromodulator systems. And then in vivo imaging. So, since my postdoc, I've been doing mostly PET imaging. We're starting to get into this, and I'll touch on it, this MR imaging of the locus coeruleus as well. And because I care about cognition, behavior is something that I hope to ground all of this work in. I was told long ago that the brain imaging can lie to you. And behavior is something that if it's working, you can really draw the conclusions that you want. So I try to link all of these, and we have ongoing projects right now in the lab that are incorporating all of these different elements, which during my initial job talks when I was on the market for an assistant professor. This was the dream that I would be doing all of this. And so now it's kind of cool to actually realize that, oh, I'm walking the walk now instead of talking the talk. Okay, so this is a recent development in the research that I'm doing, but now I'm starting to look into the locus coeruleus, and I'm going to make heretical claims about my use of PET imaging to look at locus coeruleus neurochemical function. But as you know, that's kind of a crazy thing to do because it's a teeny tiny nucleus. It's in the rostral pontine brainstem, a region that's difficult to image, in part because of the proximity of the fourth ventricle, but it's the main source of norepinephrine in the brain. It's associated with arousal, autonomic functions, and then, interesting cognitive processes like memory and executive function due to the rich innervation of the medial temporal lobe and prefrontal cortex. And then, as everyone loves to remind people, the locus coeruleus also co-releases dopamine. So that's, I tend to talk about just catecholamines in the locus coeruleus, but I should probably use more of an umbrella monoamine term. But the way that I got interested in this research, this line of research, is because it's implicated in the early pathophysiology of Alzheimer's disease. So it's one of the first regions that will accumulate abnormal tau. And I'm sure you guys are potentially sick of hearing this, because a lot of the work that's established the locus coeruleus and its implications in Alzheimer's disease has been done here by Keith Johnson and Heidi Jacobs' group and all. But I could tell you that I didn't know what the locus coeruleus was or where it was when I was in grad school, but around that time there was this pretty influential paper that came out, which was a human neuropathology paper. And what's interesting about it is that with neuropathology, you can look at different brain regions simultaneously and look at the volume and the pathology in different regions. And this was a study using the Rush data set where they had longitudinal cognitive data on individuals before they died, and then post-mortem they were able to look at all of these different subcortical structures, including the locus coeruleus, but also the VTA and the substantia nigra and the raphe nuclei, and basically pit them against one another to see, to what extent one performed better in predicting an individual's cognitive decline before they passed. And while many of these markers and the pathological markers were associated with cognitive decline, if you put them all into a model, the thing that rose to the top was the locus coeruleus. And so basically, one of the main points of this paper was that if you have high locus coeruleus cell density when you die, you're more likely to be on this more optimal aging trajectory, where in the years before you passed away, you were doing quite well. And so this was talked about as neural reserve or brain reserve, and its potential benefit in bestowing this successful maintenance of cognitive function before death. And as many of you likely know, there have been, developments and breakthroughs in MR imaging, which allow for these specialized structural scans that some people will call neuromelanin sensitive MRI. This is something that we're starting to do, but it's not really my bread and butter. But basically, this is some of our, preliminary work using these scans. But you can see this hyperintense signal in the locus coeruleus, where you reference to these pons regions in blue. And what has become quite interesting is that you'll see after the age of 60, you'll see these declines in this signal, and it seems to be a meaningful proxy for changes in cognitive function, as well as tau pathology, which I'll show you in the future. So in the future slides. And so this short-ish, like 5-minute MR scan, which is quite easy to collect. Well, it's pretty easy to collect or to tack on to your existing protocols. It's increasingly being adopted by people that are interested in aging, as well as psychopathology and other neurological disorders. And it's being added to this toolkit of potential biomarkers. The most critical one in my field, of course, in Alzheimer's disease relevant work is PIB imaging or amyloid imaging. And the great Julie Price is one of the big people in this history that we have to thank for this, for her work in the early 2000s developing it. But it allowed us to see in the living human brain the degree of pathology that people had. And so here we have an Alzheimer's patient with high binding all throughout cortex, a cognitively normal older adult. This is an amyloid negative older adult with low binding. And then this is also a cognitively normal older adult, so an amyloid positive person. So someone who we now consider is potentially on the path to Alzheimer's disease. They have substantial amyloid, but as of yet have not displayed any real cognitive decline, but we consider them preclinical Alzheimer's disease. And with sort of the spread of this or the availability of this amyloid imaging, it's pretty well established now that in most studies, about a third of cognitively normal older adults over the age of 60 will have substantial amyloid in their brain. And this has been a really interesting discovery in that it suggests that there are factors that people have potentially these cognitive reserve or resilience factors that allow them to maintain optimal or pretty good cognitive function despite pathology. And then it's also suggested that there are some people that will have substantial amyloid in their brain, and yet for some reason they're not developing tau at rates that are akin to some of their more unfortunate peers. And so there's lines of research that are interested in what the mechanisms of that are, which people have talked about as Alzheimer's disease resistance. So your ability to stave off the development of additional pathology. So when I started my postdoc, I worked with Bill Jagist, if I haven't mentioned that. He's incredible and is at UC Berkeley, and we did PET imaging up at Lawrence Berkeley National Lab. And so when I started there in about 2014, we were just starting our tau PET program with fluorotaucipir. And a lot of that early work basically demonstrated that most cognitively normal older adults, basically all cognitively normal older adults have tau pathology in the medial temporal lobe. And then if you're amyloid positive, meaning you have substantial amyloid, then you'll start to see that tau pathology in other parts of the brain and potentially spreading. All right, so just to sort of orient you about some of the work that's been done in this field linking the locus coeruleus to the spread of pathology or to the occurrence of pathology in the brain. I just want to highlight briefly this paper from Heidi Jacobs and Keith Johnson, where they basically had these MRI structural scans of the locus coeruleus. And what they found is that older adults that look like they have sort of diminished integrity of the locus coeruleus, they show higher levels of tau burden. And really in the temporal lobe. And then looking at PIB positive versus PIB negative older adults, we see that with increasing age, particularly PIB positive older adults show reduced integrity of I call it integrity, but in reduced signal in the locus coeruleus. So it seems like there's something happening specifically here with both tau and amyloid, and then there's this sort of expected interaction that comes out of that, where for people, if you have sort of an average locus coeruleus or a below average locus coeruleus, you'll see these very sort of traditional relationships where with higher amyloid, you get higher tau on the y-axis. I hope that you guys, they can probably see my cursor, I'm assuming. And then sort of one of the more interesting findings out of this paper is, again, sort of mirroring this pathology paper from Wilson, is that if you have a more intact or sort of good-looking locus coeruleus, it disrupts these relationships. So potentially one interpretation is that you can, have high amyloid, you can live with higher amyloid without having this increased tau pathology. And so this is interesting to people because it suggests that, if there's these causal relationships, that if we can do things to maintain or preserve the locus coeruleus, that this might have beneficial impacts towards decreasing potentially the spread of tau pathology. And most of these, or many researchers who are using these MR scans, it's a natural thing to do to want to map this onto the actual neurochemical health of the locus coeruleus and the norepinephrine or dopamine in particular. And we actually don't know these days, really to what extent the structural measures map onto the neurochemical health of the neuromodulator system. And this is something that we should care about because the norepinephrine, I was mentioning this earlier, has been linked to all of these sort of neuroprotective functions. So it's implicated in the maintenance of the blood-brain barrier and anti-inflammatory processes. It's important in sleep regulation. And so potentially, if you are able to maintain optimal levels of norepinephrine, this might translate into mechanisms of this resistance to pathology, like I was mentioning. And then on the flip side of that, it might also be important for the maintenance of cognitive function, so resilience or cognitive reserve as well, because we know that norepinephrine from the projections from the locus coeruleus go to these important memory regions and to prefrontal cortex, and it's implicated in all sorts of different cognitive, different aspects of cognitive performance. So we really want to be able to say something about the neurochemistry. And so I'd previously been using this PET measure, fluoro-l-m-tyrosine. So this is sort of similar to F-DOPA, which you might be familiar with. It's a measure of synthesis capacity. So throughout my postdoctoral work, we were really focused on the striatum and the high signal that we have here due to dopamine projections to the striatum. But this is a cool tracer. It was developed after F-DOPA, and the idea was that I'm not a chemist, so forgive me for my sins in describing this, but it was designed to address some of the limitations of F-DOPA in terms of its release from the synapse or from vesicles after long scanning times and all of that. So it has improved signal compared to F-DOPA, but it's a substrate for this synthesis enzyme, aromatic amino acid decarboxylase. So this is a synthesis enzyme that's involved in producing dopamine, but also norepinephrine is later down in the synthesis cascade. Separately, it's also involved in the synthesis of serotonin. So it's actually the final synthesis step in serotonin. So broadly, it's a measure of monoamine synthesis capacity. So this is actually a tracer that I'm working with Jacob Hooker right now in order to bring this online here at the Martinos Center. And so if this is at all interesting to you, I would love to talk to you about it and how we've used it in the past and what we have planned for the future. But given that it's involved in the synthesis of both dopamine and norepinephrine, I sort of asked myself, can this PET measure be used to look at the locus coeruleus? And as I mentioned before, this is sort of a heretical proposition given the size of the locus coeruleus and the spatial resolution of PET. But it was the pandemic and I wasn't collecting any data. So we started to look at the scans in the brainstem. And basically, if you look at individual subjects and these, my best examples of individual subjects, you can see something that looks like the locus coeruleus on an individual subject level in native space. And then I'm showing you these three subjects at the same threshold as what you would use to look at the striatum. And so there is a lot of signal there. And I can tell you, in some of my reading, like I've found a surprising number of PET papers that have reported locus coeruleus PET signal. And I can tell you that none of them have ever shown you what an individual subject looks like. They'll plop some sort of box or ROI over the region and then just, run their correlations. So I think, just the fact that I can stand up here and show you a few subjects that look like they have pretty good signal in the locus coeruleus is we're going in the right direction here. The issue is that the brainstem is full of monoaminergic nuclei. And so this tracer, while we get nice signal in the locus coeruleus, we get the whole brainstem is hot. So I'll just show you this is a group level map. So the good, the bad, the ugly. So we see basically signal all throughout this portion of the brainstem. And so you might say, okay, well, maybe this is the median raphe and the locus coeruleus. Maybe this is the substantia nigra and the VTA. Maybe this is the dorsal raphe. It's just unfortunate that this, while the signal is good for this tracer, it's not specific to our target of interest. And so we've been working with the data in different ways and try to address this issue. So most of our analyses will really just focus on regions of interest in the locus coeruleus, and really we've just been focusing so far a little bit on the dorsal raphe. I've never presented these data before, but I'll show you a little bit at the end. So basically, in the papers that we published on the LC so far, we'll do our main analyses on the locus coeruleus and then say, okay, well, if we do parallel analyses in the raphe, we don't see these effects, which is, not ideal, but it's how we've been handling it so far. I am interested in developing some methods for, partial volume correction, ROI-based partial volume correction using like a geometric transfer matrix approach or something like that, or adopting one. Another thing that we've looked at is basically to what extent, is all of this signal correlated across the brainstem. And so, what we've done is, extract signal from the ROIs for each participant for locus coeruleus, and then do a voxel-wise regression. So look across the whole brain, where is signal that's correlated with the signal that's within this locus coeruleus. And, in these covariance analyses, we have a data set of young subjects, and then we have a data set of older subjects. We see that it doesn't appear that it's just sort of this monotonic, hot signal that's there's no spatial selectivity to it. We do recapitulate nicely the locus coeruleus, and it doesn't seem to be highly correlated with the other brainstem regions. So permitted us to some confidence in moving forward with this. So what I'll be doing is presenting preliminary data in 49 cognitively normal older adults. These were data collected at UC Berkeley by Bill Jagus in the Berkeley Aging Cohort Study. So this, these analyses provided preliminary data for our R01, which we're starting data collection for now. So basically, we're collecting tau PET data on our participants, this Fluoro-L-m-tyrosine, so FMT, tau PET, and we're doing MR imaging to get the LC structural scan as well. And we're going to be following people longitudinally. So we'll have a lot more subjects, and we'll have longitudinal data. So we'll really be able to fill in some of these gaps here. But all of these participants have the FMT scan, an amyloid PIB scan, 14 are amyloid positive, and tau PET, which is fluorotau superior. Most of them had tau PET, 42 of the 49 participants. So, this is sort of a new realm, trying to use this PET scan in order to look at the locus coeruleus. So we did a lot of the sort of proof of concept work in the beginning. So I'm just going to show you some of our sanity checks, and potentially that will give you confidence in the data that I'll show you in a minute. But we measured tau in the meta-ROI, the JACC meta-ROI. So this is a group of temporal lobe regions that are vulnerable to tau pathology. And then looking at our PIB positive versus our PIB negative older adults, we see the expected elevation of tau in those amyloid positive preclinical AD folks. So those that we presume are on the path to Alzheimer's disease, they indeed have higher levels of tau pathology. And then we see standard relationships between temporal lobe tau and memory performance. So this is the California Verbal Learning Test. So this is like a list learning test, and cognitive assessments like this have been used in previous LC memory work, and so it seemed like an obvious choice for to sort of anchor some of our analyses. But basically what's been shown in even cognitively normal people, that higher levels of tau pathology are associated with worse memory performance. And these are really driven by the amyloid positive participants, although you'll see sometimes if your subject numbers are large enough, you'll see these effects in amyloid negative subjects as well. Okay, so that's the sanity check stuff. And one of the critical things that we're interested in looking at is what is the relationship between the catecholamine synthesis capacity in the locus coeruleus and tau pathology in the temporal lobe, accounting for PIB positive and PIB negative status. And so what we find is a significant interaction between amyloid pathology and locus coeruleus catecholamine synthesis capacity. So if you're amyloid positive and you have high levels of synthesis capacity in the locus coeruleus, we see that you are likely to have lower levels of tau pathology. So this fits very nicely with some of the previous work from the Jacobs paper that I presented to you, where they show that higher structural integrity is associated with lower tau pathology. Here we're having an actual a measure of neurochemical function, or the synthesis capacity of catecholamines, showing a complementary relationship. And I realize that there aren't so many subjects in the sample, but as an additional test of the robustness of this, we took advantage of longitudinal tau PET data that were available in these participants. So 30 of our participants had undergone longitudinal tau PET imaging. So I find these helpful, but maybe that they're kind of insane ways of plotting these data. But these are all of our individual participants that have longitudinal data. Along this line is the time at which they had FMT. This is our monoamine or catecholamine synthesis capacity measure. And then here are all of their tau PET scans in relation to that. So they're all staggered. They're kind of all over the place. But what we're able to ask is, to what extent is higher locus coeruleus catecholamine synthesis capacity associated with your tau trajectory? So are you increasing in tau at a high rate, or are you staying stable? And we find very complementary results to our cross-sectional analyses, and kind of an exciting hint that we might, with longitudinal tau PET, be able to pick up some of these associations with amyloid negative individuals. But we see that if you have substantial amyloid in your brain, but you have high levels of catecholamine synthesis capacity, you're showing less tau accumulation. It's not just lower tau at baseline, it's less accumulation. So this is I mean, obviously these are all correlational studies, but and need to be done in parallel with work in non-human animal models. But it maps on nicely with some of these theories that norepinephrine and the maintenance of optimal norepinephrine function might actually be a mechanism of resistance to pathology as well. Okay, so we didn't see direct relationships between our locus coeruleus measure and cognitive function. And so this was a little bit of a puzzle because there's been a lot established in this realm. And I just wanted to highlight some of the work and the controversy in the field of aging and Alzheimer's disease relevant work. And this is probably has parallels also in psychiatry. But in the like around 2020, there were a series of very interesting and almost like academic soap opera like opinion pieces about how we need to define terms like cognitive reserve, resilience, what is compensation, and things like that. And there was a series of white papers, opinion pieces, responses to those pieces, responses to the responses. And I don't know if we have an answer yet. My guess is that in psychiatry, there's similar things happening with resilience, because that's a word that gets thrown around a lot. But there's this concept, and I've like dropped it in some of my descriptions earlier, of this cognitive reserve, which is a property of the brain that allows for cognitive performance that is better than expected, given the degree of life course related brain changes, brain injury or disease. So they define this as like a critical interaction. So your thing of interest, so in my case, it's this norepinephrine or catecholamine measure. Does it interact with tau pathology in order to predict or in its association with cognitive function? So can you produce like better than expected cognitive results, given the level of pathology? And we, in our research, and a lot of this research has been done here, is that we've been talking about cognitive reserve proxies for a long time in aging. So in years of education, what you achieved in terms of your occupation, your IQ, all of these things are associated with better than expected cognitive function given brain insults, given your atrophy and all of that. And they're useful proxies, but it would be nice to be able to link them to the brain mechanism. So what is it about these things that's imbuing these benefits to cognitive function? And so I think that neuromodulatory systems are a compelling mechanism for cognitive reserve because of how they input to all of these regions of interest in cortex and in subcortical regions, and they're already implicated in memory and executive function and all of these things. So indeed, oopsies, I forgot how I go forward in my slides. Indeed, I'm gonna turn this off, I guess. I don't know if I can actually. Yeah. I'm still not seeing.
Yeah, you can. Under the bar. So yeah, you maybe just disable it. Yeah.
I would just.
Yeah.
Yeah.
Let's see if this setting still worked. Oh God. Our favorite. Okay, let's see if I have on memory. All right. Is it on our video? Okay, cool. We've escaped. Okay, so indeed we do see this interaction that people ask for when you say something might be cognitive reserve. So here, sorry, these are just the model fits without the individual subject data on them. But here, we have tau pathology on the x-axis, and then on the y-axis, we have that same memory measure that I showed you before. So in people that have low catecholamine synthesis, so non-optimal, we see this sort of standard relationship where higher levels of tau are associated with worse memory. But this relationship, it seems to be disrupted if you have high catecholamine synthesis capacity in your locus coeruleus. So this is the better than expected memory performance given tau. So you could potentially have, higher tau burden, but still be maintaining.
Why is that slope positive? That's hard.
Yeah, well, it's yeah.
But indeed,
This is the issue with the subjects, the number of subjects that we have. So basically, we're splitting. The critical interaction is significant, but it's hard to power these well with, it has to be a really, strong, large effect size in order to be adequately powered. So I'm curious to see whether this is going to hold when we're analyzing closer to 100 participants. Yeah, you'll see similar stuff with some of the IQ proxies as well, going in funky directions. Okay, and then, as I was saying, that because of sort of the novelty of this and the small number of participants that we have, another sort of test of this is to look at the relationship between locus coeruleus catecholamine synthesis capacity and its relationship to another proxy or a proxy for cognitive reserve. So what we had in this data set is the lifetime experiences questionnaire. So this asks questions about the level of educational attainment, your job, what was required of you in your job, how much management responsibility you had. So there's potentially a social component to that, what your activities have been since you've retired, and then your physical activity across the lifespan. So it's kind of it's an interesting measure. It's kind of hard to score and to walk people through it. But what we're finding with it is, independent of amyloid status, that there is this positive relationship between catecholamine synthesis capacity and your performance on the lifetime experiences questionnaire. So like how sort of intellectually and physically enriched your life has been. And there's some work in animal models that suggests, if you take a rat and you expose it to an enriched environment, expose it to lots of novelty, that you will see changes in the norepinephrine system. And so it's possible that there is, my guess is that these are all bidirectional relationships, but it is possible that this environmental enrichment in these results are really driven by people's self-reported activity in middle age. So it is possible that these activities in middle age might actually help boost norepinephrine. The question is, do those effects last more than a week? Who knows? Okay, so this is just a quick interim summary. So this is sort of this dive that we've been taking into trying to establish whether our PET measure of locus coeruleus really can fit in with this broader literature that has been driven by neuropathology data and now MR imaging. And we are finding evidence that fits nicely with the established work where higher levels of catecholamine synthesis are associated with lower tau and less accumulation of tau pathology, and then suggestions that this might be a useful measure to think about when considering cognitive reserve and cognitive reserve mechanisms. And so this is, let me just check the time. We're doing pretty well. So we've basically to date been totally ignoring signal in the raphe nuclei. We'll do these control analyses and demonstrate that the effects that we demonstrate with the locus coeruleus aren't true in the raphe, but we're just starting now to look more seriously at the dorsal raphe. So I'll just remind you of what the signal looks like, and basically using a region of interest approach to extract signal from this region. And we do see, sort of similar things as our locus coeruleus covariance analyses, where it's not signal in the dorsal raphe isn't highly correlated with signal in other brainstem regions. So we decided to start looking at the dorsal raphe because it also has some possible connections to the pathophysiology to Alzheimer's disease. And it is a powerful, if you can sort of assess multiple systems in adding those to your models, and you're collecting the data anyway, it's potentially quite illuminating. In the similar way to the work in Wilson in that neuropathology paper, where he was able to, at the same time, look at locus coeruleus cell density as well as dorsal raphe cell density, and see which one is about, like if you're accounting for the other, which predicts cognitive trajectories. It's potentially quite interesting. So similar to the locus coeruleus, the dorsal raphe develops abnormal tau before the entorhinal cortex. It's largely ignored, but Leah Greenberg at UCSF and a lot of her colleagues have done important work in this area. And then there's some indirect evidence also linking the dorsal raphe to Alzheimer's disease. So monoamine systems, including serotonin, have been implicated in depression, and we know that increasing depression symptoms are considered by many to be a prodromal stage of dementia. And then there's this sort of other interesting observation that women are at greater risk of developing both depression and Alzheimer's disease. And so looking at the raphe nucleus in the serotonin system might be an interesting region where you might get at some of those intersections between Alzheimer's disease related pathology and mechanisms of psychopathology, including depression. So one way in which the dorsal raphe nuclei are different from the locus coeruleus is that we have whopping age effects in the dorsal raphe. So we have a group of young subjects that have gotten the Fluoro-L-m-tyrosine scan, and then older subjects. And we see, just doing a region of interest-based analysis, we see much higher signal in older subjects. And this is opposite to what you would expect if these were driven by partial volume effects due to age. And then even like on a voxel-wise level, doing a t-test, older versus young, you see higher signal in a region that looks much like the dorsal raphe. So this is the region of interest that we've been using in our analysis, just overlaid here. And so this fits in with some other, a growing body of research suggesting that there's upregulation of activity and potentially expression of synthesis enzymes across the lifespan. We see very similar things in the dopamine system in the striatum. So there have been a number of meta-analyses that have suggested that other markers, transporters, receptors will decline, but it seems like the synthesis capacity or the enzymes are maintained, or depending on who you're talking to, may be upregulated. And you'll see this in animal models as well, lesioning a target region or lesioning the nucleus itself will lead to upregulation of the expression of synthesis enzymes and also their activity. Okay, so basically we've continued on looking at just basic demographic relationships. So here I'm showing you young adults and older adults. This is their signal in the dorsal raphe nucleus. We don't see sex differences in young adults. Overall, they just have lower signal than older adults. But in our older adults, we do see a significant increase in the signal for females relative to males. So this is in line with some of the original interest in this region as a possible target of looking at these interactions between sex and psychopathology and pathology. Okay, so to investigate the dorsal raphe and what it's associated with, again, one of these plots that I find helpful but might not be helpful to everyone. We took advantage of longitudinal MRI, structural MRI imaging that we had on these participants, as well as longitudinal cognitive testing where we collected the geriatric depression scale, as well as we had PIB, amyloid PET scans, and then tau PET scans for all of these participants as well. And then in the black line, that's where the FMT scan occurred. So that's what we're using to measure the dorsal raphe monoamine synthesis capacity. And so basically we're interested in what does this elevation in raphe signal mean? Is it a signal of positive aging trajectories, negative aging trajectories, like what's going on here? So we started by looking at the longitudinal MRI scan, so looking at changes in cortical thickness. And what we're finding is that those people with higher levels of the raphe FMT signal, they're the ones that are showing greater atrophy. And in particular in this region of the banks of the superior temporal sulcus, so in this temporal lobe region. And we didn't see any regions where like thicker or like the maintaining of cortex was associated with higher with higher signal. So it seems like we're seeing this elevation in signal in the context of greater atrophy. Unlike the locus coeruleus, we don't see direct relationships between our raphe signal and any of our amyloid or tau pathologies or any interactions. So it seems like the link potentially to the mechanisms of Alzheimer's disease, if you believe these data, are much more tenuous. We're not seeing those direct relationships. And we don't see any direct relationships with depression symptoms cross-sectionally or longitudinal change in depression symptoms. So again, we sort of, in trying to think about possible modification, like interactions that might suggest that, one sort of tempting conclusion is that there's a compensatory upregulation of activity of amino acid decarboxylase in response to atrophy. And so, one of our questions is there any evidence that suggests that those subjects that have higher levels of synthesis are, doing better at all? And we don't see evidence for that. This is a plot just showing relationships between tau pathology and longitudinal slope of the geriatric depression scale. So higher scores here mean that you're increasing in depression symptoms over time. And so, some groups with larger sample sizes are showing, overall like main effects, positive relationships between tau pathology and depression symptoms. We don't see that in this data set. We see it in women. So women with higher entorhinal cortex tau pathology, this is actually true of other regions like amygdala and in the Jack meta ROI that I showed you previously. But it seems to be something that at least in this sample is specific to women. And then testing this sort of critical interaction between the PET signal in the dorsal raphe, it interacts with tau pathology. So what we're seeing is that those people with the highest levels of monoamine synthesis capacity in the raphe are showing these positive relationships where higher levels of tau pathology are associated with increasing depression. Oops. So it suggests that yeah, so this is adjusting for it survives adjusting for sex, but I have to imagine that we know that more of the women in the sample have higher DRN. Yeah, but it's something that we weren't well powered to look at this in this sample, but it's something that we have planned for the future. Yeah, so we see no evidence that, having higher levels of serotonin synthesis is associated with, any sort of like protection or, resilience, affective resilience or anything like this. And obviously this is a developing line of work, but it seems like an interesting way to probe potential interactions between pathology and psychopathology that's particularly relevant to women's vulnerability to both of these syndromes. So just to summarize, FMT measures of the locus coeruleus, they complement developing lines of MR research. And I think that they do offer direct insight into the neurochemical function of this region and is useful for models of Alzheimer's disease resistance and resilience. The raphe nuclei don't show these direct relationships with Alzheimer's disease pathology, but elevated monoamine synthesis capacity appears to signal poorer aging trajectories. And so our ongoing research aims to improve our methods for ROI-based partial volume correction. And then longitudinally, we're going to track changes in signal across these regions, in addition to changes in tau pathology, in addition to changes in affect and memory and all of that in our cognitively normal older adults. So with that, I just wanted to acknowledge Bill Jagus, who's has been an incredibly generous mentor and enabling me to do heretical things with the PET data that we collected together. And then the work that I'm presenting was the work of two graduate students, Claire and Jordan, who published our locus coeruleus papers. And then Teddy has been developing the line of investigation into the raphe nucleus. And then Jacob Hooker is my lifeline here and has enabled my continuing this research sort of successfully. Right, thank you.
Thank you for a very interesting talk, Dr. Berry. I would like to remind our audience to address your question by raising your hand or texting in the Q&A box. Have any questions from the audience? Yes, please.
If you go back to that locus coeruleus data where you showed the cognitive effects where you had that positive slope with the high TMC synthesis rates.
For the locus coeruleus?
Yeah, well, why?
This one.
Yeah, right there, that one on the left. It kind of, so the high rate has inferior cognitive performance in cases where they have low tau height. So is that a true statement, or is that not a significant statement?
So are you saying, is this really a positive slope?
Never mind about the slope itself, but just go to the low, tau binding point, right? You have a low tau binding point.
Yeah.
And if you look there, the performance of the people with the high synthesis rates seems to be much inferior to those that have very low synthesis rates.
Oh, yeah. So unfortunately, I feel.
Is that just unpopulated by a lot of subjects?
Yeah, I think this is like a sparsity issue, to tell you the truth. I mean, like what would make, in a perfect world that we would be, in a relationship like this. But, this is the real disadvantage of the stupid plot that I have that doesn't have any of the data points on it. So I apologize for that.
Oh, yeah, I mean, it could just be sparsely populated in some slopes.
Yeah, it's not something that we've interpreted too much.
I mean, you were kind of making the point that there was a nice correlation between the cognitive reserve and the synthesis rates, and yet that would seem that there's something weird going on.
Yeah.
And so this would kind of parallel the raphe, which is that, you have the high synthesis rate of the raphe, then you have a worse outcome. And this would be a high synthesis rate of a worse outcome.
I think that this is like not a true like I don't think there's anything significant about the slope there of that subset of the data. But yeah, no, I think considering like it's still early days, but I am, interested in considering the ways in which it's a double-edged sword. I mean, one thing, okay, I'll just tell you like the sort of history of this PET tracer and the work that I was doing as a postdoc was looking at the striatal signal. And, similarly in the striatum, you have this apparent upregulation of activity of this enzyme. So we see, and we've replicated this 3 times in 3 different data sets, higher synthesis capacity of dopamine in the striatum in older people compared to young people. And for the most part, these are older people that aren't doing very well. So they show worse cognitive performance, like just correlationally worse cognitive performance. They also show more atrophy as well. And so it was this sort of perplexing, thing in the work that we were doing where it seems like these higher levels of dopamine, are signaling like, poorer cognitive performance and like brain health in general. But despite that, you see these interactions between the atrophy that these older adults are experiencing and their level of synthesis capacity in the striatum. And actually, while overall they're doing worse, if you have very high levels of synthesis capacity, it does seem like there is still this resilience or this reserve mechanism. So despite the higher levels of atrophy, they're able to maintain. We're not looking at episodic memory for those subjects. We're looking more at like working memory, executive function measures, but they'll show like maintenance. Those people that are synthesizing the most will show sort of maintenance of working memory over time. So it does seem like there might be some like, subtle effect that it is beneficial to have higher levels of synthesis capacity, but overall, it's associated with poor aging trajectory. So it's a complicated.
Kind of like what happened. Basically, the target and the synthesis, are suffering perhaps differential temporal trajectories with regards to generation.
Right.
Give people L-dopa to ramp up the synthesis of the remaining cells because the targets are becoming sparser.
Mm-hmm.
So maybe here, the upregulated synthesis rate is just a reflection of the fact that there's fewer targets. So you're kind of trying to populate more to get these defined.
Yeah. No, I think it's a great idea. We in some of these subjects, we're going to have raclopride data on them too. So I'm hoping that we can ask that, like looking at pre and post synaptic measures in our older participants. And there's actually PET work in Parkinson's that's shown that that you will see higher levels of they use F-dopa, but higher levels of dopamine synthesis in Parkinson's. And then in longitudinal scanning, that ends up going down after a while because you just can't maintain that over too long. But yeah.
And there's also the possibility that maybe just because you're uptaking more of the substrate, it doesn't necessarily mean that it's actually synthesized.
Yeah, exactly. And in young people, we've done imperfect studies where we look at the synthesis capacity using this Fluoro-L-m-tyrosine and then doing the raclopride to scan like dopamine release measure with methylphenidate. And we see relationships between receptor density at baseline and synthesis, positive relationships like higher density of receptors with higher synthesis capacity, but we're not seeing any relationship with potentially an imperfect measure of endogenous dopamine release. But yeah, the PET imaging is so interesting because like you want to make these conclusions about the measures that you're looking at, but you have only like one little fraction of the picture in a highly dynamically regulated system, right? Where there's changes in receptors that can be very dynamic as well.
In the interest of time, we're closing the session now, but for in-person audience, you can still ask her the question after the talk. Thank you for coming for us, giving us great talk, Dr. Berry, and thank you for everyone to join us today.